Hello, my name is Sammie Haskin, and I am a student in the master's program at Kennesaw State University in Applied Statistics and Data Science. I am also an avid learner on the Coursera platform, in which I have learned many machine learning techniques as well as deep learning techniques that could very much so prove useful in a variety of fields. In today's study, I attempt to make clear the possible uses of deep learning and machine learning in the medical field. In this experiment, I sought to utilize image classification techniques to distinguish cancerous cell tissue from healthy cell tissue. The implementation of a successful cancer treatment hinges on correct diagnoses, proper treatment protocols prescribed by the doctors, and timely treatment protocols prescribed precisely when a cancer is in its early stages. Artificial intelligence can thus play a pivotal role in the accurate identification of cancer. Within this analysis, we sought to create an algorithm that could reliably classify cell tissue as benign or cancerous. To do so, we implemented two key learning methods. The first being machine learning, or the process of utilizing algorithms for the purposes of discovering patterns within data and the conduction of classification, clustering, or prediction. In deep learning, we seek to augment the capabilities of machine learning via the use of neural networks that simulate the capacities of the human brain to learn. In the works of Borkowski and others in 2019, we utilized images of benign and cancerous lung colon cell tissue for the purposes of classification using machine and deep learning algorithms. The resulting dataset contained two features. First, the image as measured as pixel intensities and colors on a scale from 0 to 256 in integers. We also saw the measurements of whether each cell was classified as benign lung tissue, benign colon tissue, lung squamous cell carcinoma, colon adenocarcinoma, or lung adenocarcinoma. The images of histopathological cell tissue were similarly measured, with a high quality of 768 by 768 pixels as saved in JPEG files. These files all exhibited a high degree of quality for use in machine learning and deep learning. Exploring the data, we see that it was derived from various sources by Borkowski in 2019, in which the original dataset contained 250 images for each class of 5 different cell types. Utilizing image augmentation, Borkowski increased that total from 250 images per category to exactly 25,000 images per category. Using these data, we can create an algorithm that could potentially be useful in the prediction of whether a cell is cancerous or not. In other words, improve the diagnostic capabilities of the medical system. Within this analysis, the Python programming language was used. We first sought to perform the extraction transform loading process, in which images were retrieved from the repository of Borkowski in 2019. These images were subsequently read into batches iteratively at a time as 100 by 150 images across a scale of red, blue, and green color channels. As these images were read in batches, the process of modeling was performed without the necessity to load all images at one time into memory, as such could have otherwise overloaded the memory of my current computer. With the Keras image generator, 2 utilized models were subsequently created, trained, and evaluated. The machine learning procedure of multiple logistic regression was first implemented using the Keras API. Using this algorithm, we were able to classify observations into one of multiple classes with an established measure of doing so. The formula for the multiple logistic regression model is shown below. For the purposes of comparison of this model to a deep learning model, a convolutional neural network was created using the same Keras API. CNNs are generally accepted as extremely effective for classifications of images, as these algorithms utilize special procedures for the purposes of reading data similar to how the human brain classifies visual stimuli. In the evaluation of the capabilities of each model to be utilized as a means of prediction of cell types, 20,000 images were utilized to train each model, while 5,000 images were utilized to test each model. For the comparison of these two models, the accuracy and F1 score was then evaluated and prepared. For the purposes of evaluating the model, we sought to first calculate the precision of each model, which is the number of true positives over the number of all true positives and false positives, while recall is the number of true positives over all the specific instances of one class. The F-score is calculated below using the precision and recall. In the evaluation of the efficacy of the logistic regression model, we saw that this model resulted in an accuracy score of 40.65 on the data. This means that accurate predictions resulted 40.65% of the time on the data that the algorithm was trained on. When observing the accuracy of the same model on testing data, we see that it was not as effective, as it only predicted 29.70% of all classifications as the correct classification for the cell type. Similarly, this model resulted in an F1 score of 0.2975. This indicates that this model only predicted slightly better than chance, and indicates that this model was nowhere near optimal for use in clinical settings. In the evaluation of the convolutional neural network, a high degree of accuracy on the training data was indicated with a score of 94.45. When exposed to the remaining 5,000 observations, we saw that the algorithm was extremely effective and generalized well, with the accuracy score of 92.0 and an F-score of 0.9210. These metrics seemed to indicate that the model generalizes well to new data points, and that this algorithm could be relatively useful when implemented in clinical settings. In summary, the deep learning model was significantly accurate in its classification of each tissue as benign or cancerous. The logistic regression procedure alone, however, could not produce the same degree of accuracy as produced by the deep learning model. The convolutional neural network, however, generalized well to new data points and indicates promise when used in clinical settings for the classification of histopathological images of lung or colon cell tissue, and could greatly aid in future diagnoses. Through the use of convolutional neural networks, the efficacy of the classification of images in a variety of fields can be greatly increased. The effectiveness of these procedures was illustrated in this project through the classification of colon and lung cell tissue as benign or cancerous. With more data, these models could potentially be generalized to a variety of different illnesses to aid classification of different pathologies for doctors. Once again, much credit goes to Borkowski in 2019, as they created a data set that exhibited high reliability. So once again, thank you all for watching this presentation, and I hope you have learned a lot as it pertains to the uses of artificial intelligence in the medical field. Thank you, and bye-bye.